A leaf is an organ of a vascular plant and is the principal lateral appendage of the stem. The leaves and stem together to form the shoot. Foliage is a mass noun that refers to leaves collectively. Typically a leaf is a thin, dorsoventrally flattened organ, borne above ground and specialized for photosynthesis. Most leaves have distinctive upper and lower surfaces that differ in color, hairiness, the number of stomata and other features. In most plant species, leaves are broad and flat. Such species are referred to as broad-leaved plants. Many gymnosperm species have thin needle-like lobes that can be advantageous in cold climates frequented by snow and frost. Leaves can also have other shapes and forms such as the scales in certain species of conifers. Some leaves are not above ground. Succulent plants often have thick juicy leaves, but some leaves are without major photosynthetic function and may be dead at maturity, as in some cataphyls and spines. Furthermore, several kinds of leaf-like structures found in vascular plants are not totally homologous with them. Examples include flattened plant stems, and phyllodes, both of which differ from leaves in their structure and origin. Many structures of non-vascular plants, and even of some lichens, which are not plants at all, look and function much like leaves. The primary site of photosynthesis in most leaves almost always occurs on the upper side of the blade or lamina of the leaf, but in some species, including the mature foliage of Eucalyptus palisade, occurs on both sides and the leaves are said to be isobilateral. Leaf development. According to Agnes Arber's partial shoot theory of the leaf, leaves are partial shoots. Compound leaves are closer to shoots than simple leaves. Developmental studies have shown that compound leaves, like shoots, may branch in 3 dimensions. On the basis of molecular genetics, Eckard and Baum concluded that, it is now generally accepted that compound leaves express both leaf and shoot properties, general characteristics of leaves. Typically leaves are broad, flat and thin, thereby maximizing the surface area directly exposed to light and promoting photosynthetic function. They are arranged on the plant so as to expose the surfaces to light as efficiently as possible without shading each other. But there are many exceptions and complications. For instance, plants adapted to windy conditions may have pendant leaves, such as in many willows and eucalyptus. The internal organization of most kinds of leaves has evolved to maximize exposure of the photosynthetic organelles, the chloroplasts, to light and to increase the absorption of carbon dioxide. Gas exchange is controlled by stomata, which open or close to regulate the exchange of carbon dioxide, oxygen, and water vapor with the atmosphere. In a given square centimeter of a plant leaf there may be from 1,000 to 100,000 stomata. Some leaf forms are adapted to modulate the amount of light they absorb to avoid or mitigate excessive heat, ultraviolet damage, or desiccation, or to sacrifice light absorption efficiency in favor of protection from herbivory. For xerophytes the major constraint is not light flux or intensity, but drought. Some window plants such as Fenestraria species and some Howwithia species such as Howwithia tessellata and Howwithia truncata are examples of xerophytes. And Bulbemus embryanthymoides. The shape and structure of leaves vary considerably from species to species of plant, depending largely on their adaptation to climate and available light, but also to other factors such as grazing animals, available nutrients, and ecological competition from other plants. Considerable changes in leaf type occur within species too. For example, as a plant matures, as a case in point, eucalyptus species commonly have isobilateral, pendant leaves when mature and dominating their neighbors. However, such trees tend to have erect or horizontal dorsoventral leaves as seedlings, when their growth is limited by the available light. Other factors include the need to balance water loss at high temperature and low humidity against the need to absorb atmospheric carbon dioxide. In most plants, leaves also are the primary organs responsible for transpiration and guttation. Leaves can also store food and water, and are modified accordingly to meet these functions. For example, in the leaves of succulent plants and in bulb scales. Accordingly, leaves are prominent in the diet of many animals. This is true for humans, for whom leaf vegetables commonly are food staples. Correspondingly, leaves represent heavy investment on the part of the plants bearing them, and their retention or disposition are the subject of elaborate strategies for dealing with pest pressures, seasonal conditions, and protective measures such as the growth of thorns and the production of phytoliths, lignans, tannins and poisons. Deciduous plants in frigid or cold, temperate regions typically shed their leaves in autumn, whereas in areas with a severe dry season, some plants may shed their leaves until the dry season ends. In either case, the shed leaves may be expected to contribute their retained nutrients to the soil where they fall. In contrast, many other non-seasonal plants, such as palms and conifers, retain their leaves for long periods, while witchia retains its two main leaves throughout a lifetime that may exceed 1,000 years. Not all plants have true leaves. Bryophytes are non-vascular plants, and, although they produce flattened, leaf-like structures that are rich in chlorophyll, these organs differ morphologically from the leaves of vascular plants. For example, they lack vascular tissue, are usually only a single cell thick, and lack an internal system of intercellular spaces. Simple, vascularized leaves first evolved in club mosses during the Silurian period, but true leaves or euphyls of larger size and with more complex venation did not become widespread in other groups until the Devonian period, by which time the carbon dioxide concentration in the atmosphere had dropped significantly. This occurred independently in several separate lineages of vascular plants, the Sphenopsida, ferns, and progymnosperms, and later in the gymnosperms and angiosperms. Euphyls are also referred to as macrophyls or megaphylls. Large-scale features. A structurally complete leaf of an angiosperm consists of a petiole, a lamina, and stipules. Not every species produces leaves with all of these structural components. In certain species, paired stipules are not obvious or absent altogether. A petiole may be absent, or the blade may not be laminar. The tremendous variety shown in leaf structure from species to species is presented in detail below under Morphology. The petiole mechanically links the leaf to the plant and provides the route for transfer of water and sugars to and from the leaf. The lamina is typically the location of the majority of photosynthesis. The upper angle between a leaf and a stem is known as the axil of the leaf. It is often the location of a bud. Structures located there are called axillary. Anatomy. Medium-scale features. Leaves are normally extensively vascularized and typically have networks of vascular bundles containing xylem, which supplies water for photosynthesis, and phloem, which transports the sugars produced by photosynthesis. Many leaves are covered in trichomes which have diverse structures and functions. Small-scale features. The major tissue systems present are the epidermis, which covers the upper and lower surfaces, the mesophyll tissue inside the leaf, which is rich in chloroplasts, the arrangement of veins. These three tissue systems typically form a regular organization at the cellular scale. Specialized cells that differ markedly from surrounding cells, and which often synthesize specialized products such as crystals, are termed idioblasts. Major leaf tissues. Cross-section of a leaf. Epidermal cells. Spongy mesophyll cells. Epidermis. The epidermis is the outer layer of cells covering the leaf. It is covered with a waxy cuticle which is impermeable to liquid water and water vapor and forms the boundary separating the plant's inner cells from the external world. The cuticle is in some cases thinner on the lower epidermis than on the upper epidermis, and is generally thicker on leaves from dry climates as compared with those from wet climates. The epidermis serves several functions: protection against water loss by way of transpiration, regulation of gas exchange, secretion of metabolic compounds, and absorption of water. Most leaves show dorsoventral anatomy. The upper and lower surfaces have somewhat different construction and may serve different functions. The epidermis tissue includes several differentiated cell types: epidermal cells, epidermal hair cells, cells in the stomatal complex, guard cells and subsidiary cells. The epidermal cells are the most numerous, largest, and least specialized and form the majority of the epidermis. These are typically more elongated in the leaves of monocots than in those of dicots. Chloroplasts are generally absent in epidermal cells, the exception being the guard cells of the stomata. The stomatal pores perforate the epidermis and are surrounded on each side by chloroplast containing guard cells, and 2 to 4 subsidiary cells that lack chloroplasts, forming a specialized cell group known as the stomatal complex. The opening and closing of the stomatal aperture is controlled by the stomatal complex and regulates the exchange of gases and water vapor between the outside air and the interior of the leaf. Stomata therefore play the important role in allowing photosynthesis without letting the leaf dry out. In a typical leaf, the stomata are more numerous over the abaxial epidermis than the adaxial epidermis and are more numerous in plants from cooler climates. Mesophyll. Most of the interior of the leaf between the upper and lower layers of epidermis is a parenchyma or chlorenchyma tissue called the mesophyll. This assimilation tissue is the primary location of photosynthesis in the plant. The products of photosynthesis are called assimilates. In ferns and most flowering plants, the mesophyll is divided into 2 layers. An upper palisade layer of vertically elongated cells, 1 to 2 cells thick directly beneath the adaxial epidermis, with intercellular air spaces between them. Its cells contain many more chloroplasts than the spongy layer. These long cylindrical cells are regularly arranged in 1 to 5 rows. Cylindrical cells, with the chloroplasts close to the walls of the cell, can take optimal advantage of light. This light separation of the cells provides maximum absorption of carbon dioxide. This separation must be minimal to afford capillary action for water distribution. In order to adapt to their different environment, plants had to adapt this structure to obtain optimal result. Sun leaves have a multi-layered palisade layer, while shade leaves or older leaves closer to the soil are single-layered. Beneath the palisade layer is the spongy layer. The cells of the spongy layer are more branched and not so tightly packed, so that there are large intercellular air spaces between them for oxygen and carbon dioxide to diffuse in and out of during respiration and photosynthesis. These cells contain fewer chloroplasts than those of the palisade layer. The pores or stomata of the epidermis open into substomatal chambers, which are connected to the air spaces between the spongy layer cells. Leaves are normally green, due to chlorophyll in chloroplasts in the chlorenchyma cells. Plants that lack chlorophyll cannot photosynthesize. Veins. The veins are the vascular tissue of the leaf and are located in the spongy layer of the mesophyll. The pattern of the veins is called venation. In angiosperms, the venation is typically parallel in monocotyledons and forms an interconnecting network in broad-leaved plants. They were once thought to be typical examples of pattern formation through ramification, but they may instead exemplify a pattern formed in a stressed tensor field. A vein is made up of a vascular bundle. At the core of each bundle are clusters of 2 distinct types of conducting cells. Xylem, cells that bring water and minerals from the roots into the leaf. Phloem, cells that usually move sap, with dissolved sucrose, produced by photosynthesis in the leaf, out of the leaf. A sheath of ground tissue made of lignin surrounding the vascular tissue. This sheath has a mechanical role in strengthening the rigidity of the leaf. The xylem typically lies on the adaxial side of the vascular bundle and the phloem typically lies on the abaxial side. Both are embedded in a dense parenchyma tissue, called the sheath, which usually includes some structural collenchyma tissue. Seasonal leaf loss. Leaves in temperate, boreal, and seasonally dry zones may be seasonally deciduous. This mechanism to shed leaves is called abscission. When the leaf is shed, it leaves a leaf scar on the twig. In cold autumns, they sometimes change color, and turn yellow, bright orange, or red, as various accessory pigments are revealed when the tree responds to cold and reduced sunlight by curtailing chlorophyll production. Red anthocyanin pigments are now thought to be produced in the leaf as it dies, possibly to mask the yellow hue left when the chlorophyll is lost. Yellow leaves appear to attract herbivores such as aphids. Optical masking of chlorophyll by anthocyanins reduces risk of photooxidative damage to leaf cells as they senesce, which otherwise may lower the efficiency of nutrient retrieval from senescing autumn leaves. Terminology: shape-edge ciliate, fringed with hairs; crenate, wavy-toothed, dentate with rounded teeth; crenulate, finely or shallowly crenate; dentate, toothed, such as castanea, coarse-toothed or coarsely dentate, with large teeth; glandular-toothed or glandularly dentate, with teeth that bear glands; denticulate, finely toothed, doubly toothed, each tooth bearing smaller teeth, such as ulmus; entire, even, with a smooth margin, without toothing; linear, parallel margins, elongated; lobate, indented, with the indentations not reaching to the center, such as many quercus; palmately lobed, indented, with the indentations reaching to the center, such as humulus; serrate, sawtoothed, with asymmetrical teeth pointing forward, such as urtica; cerulate, finely serrate; sinuate, with deep, wave-like indentations; coarsely crenate, such as many rumex; spiny or pungent, with stiff, sharp points, such as semilex and cirsium; tip acuminate, long-pointed, prolonged into a narrow, tapering point in a concave manner; acute, ending in a sharp, but not prolonged point; cuspidate, with a sharp, elongated, rigid tip, tipped with a cusp; emarginate, indented, with a shallow notch at the tip; mucronate, abruptly tipped with a small short point, as a continuation of the midrib, tipped with a mucronulate, mucronate, but with a noticeably diminutive spine; a mucronule; obcordate, inversely heart-shaped, deeply notched at the top; obtuse, rounded or blunt; truncate, ending abruptly with a flat end that looks cut off; base acuminate, coming to a sharp, narrow, prolonged point; acute, coming to a sharp, but not prolonged point; auriculate, ear-shaped; cordate, heart-shaped with the notch towards the stalk; cunita, wedge-shaped; haystate, shaped like an halberd and with the basal lobes pointing outward; oblique, slanting; reniform, kidney-shaped but rounder and broader than long; rounded, curving shape; sagittate, shaped like an arrowhead and with the acute basal lobes pointing downward; truncate, ending abruptly with a flat end that looks cut off; surface coriaceous, leathery, stiff and tough but somewhat flexible; farinos, bearing farina, mealy, covered with a waxy, whitish powder; glabrous, smooth, not hairy; glaucous, with a whitish bloom, covered with a very fine, bluish-white powder; glutinous, sticky, viscid, lepidote-coated with scurfy scales, used particularly of rhododendron; papillate, or papillose, bearing papillae; pubescent, covered with erect hairs; punctate, marked with dots, dotted with depressions or with translucent glands or colored dots; rugose, deeply wrinkled, with veins clearly visible; scurfy, covered with tiny, broad scale-like particles; tuberculate, covered with tubercles, covered with warty prominences; verrucose, warted, with warty outgrowths; viscid, or viscous, covered with thick, sticky secretions. The leaf surface is also host to a large variety of microorganisms. In this context it is referred to as the phyllosphere. Hairiness. Hairs on plants are properly called trichomes. Leaves can show several degrees of hairiness. The meaning of several of the following terms can overlap: arachnoid, or arachnose, with many fine, entangled hairs giving a cobwebby appearance. Barbillate, with finely barbed hairs. Bearded, with long, stiff hairs. Bristly, with stiff hair like prickles. Canescent, hoary with dense grayish-white pubescence. Ciliate, marginally fringed with short hairs. Ciliolate, minutely ciliate. Floccose, with flocks of soft woolly hairs which tend to rub off. Glabrescent, losing hairs with age. Glabrous, no hairs of any kind present. Glandular, with a gland at the tip of the hair. Hirsute, with rather rough or stiff hairs. Hispid, with rigid, bristly hairs. Hispidulous, minutely hispid. Hoary, with a fine, close grayish-white pubescence. Lanate, or lanose, with woolly hairs. Pilose, with soft clearly separated hairs. Puberulent, or puberulous, with fine, minute hairs. Pubescent, with soft short and erect hairs. Scabrous, or scabrid, rough to the touch. Cercious, silky appearance through fine, straight and oppressed hairs. Silky, without pressed, soft and straight pubescence. Stellate, or stelliform, with star-shaped hairs. Strigose, with oppressed, sharp, straight and stiff hairs. Tomentose, densely pubescent with matted, soft white woolly hairs. Carnotomentose, between canescent and tomentose. Felted tomentose, woolly and matted with curly hairs. Tomentulose, minutely or only slightly tomentose. Villous, with long and soft hairs, usually curved. Woolly, with long, soft and tortuous or matted hairs. Timing hysterant house, developing after the flowers. Cinnamon house, developing at the same time as the flowers. Patterning maculate, stained, spotted. Compare immaculate. Venation channeled, sunken below the surface, resulting in a rounded channel.